Okay, good afternoon, everybody. Welcome to today's Coffee Science Seminar. Our speaker today is Sarah Jacob, but I'll introduce her more formally in a minute. So if we go to the next slide, please, Sarah. I'm Mike Gidley. I'm Emeritus Professor in the Centre for Nutrition and Food Science. It is Coffee. And it's my privilege to be your chair for today. So I'll give you the introduction and then I'll come back at the end and facilitate the Q&A session. Next slide, please. So the University of Queensland acknowledges the traditional owners and their custodianship of the lands on which we meet. And we pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. Next slide, please. So today's seminar is scheduled for until 1 o'clock. Feel free to put any questions down during the seminar. Please use the Q&A tab at the bottom of your screen and not the chat tab. We won't be looking at the chat function. We will only be looking at the Q&A tab for any questions. You'll have another chance to book, well, you can book questions in at any time during the talk or even after the talk is finished as they occur to you. And then I will moderate those questions and put them to Sarah at the end of that. Okay, next slide, please. So today's speaker is Sarah Jacob. Sarah is a final year PhD student. I've had the privilege of being her principal advisor, and it's great to see the story coming to a really interesting set of conclusions. So Sarah's going to tell you about that journey and particularly emphasize the relationships between modern food science and traditional knowledge and the opportunities that come when you put those two alongside each other. And so Sarah will introduce herself to you as she begins her presentation. So I think I'll hand over to you now, Sarah. Take it away.
Thank you, Mike. So I would also like to begin with an acknowledgement of country. I acknowledge the traditional owners and their custodianship of the lands on which we meet today and where the seeds for this project have been sourced. I pay my respects to their ancestors and their descendants who continue cultural and spiritual connections to country, and I recognize their valuable contributions to Australian and global society. So before we begin, like Mike said, I just wanted to give a brief introduction about myself. My name is Sarah Susan Jacob, and I come from a small state in the south of India called Kerala. And some of our native produce that is integral to our diet and our lifestyle include coconut, curry leaves, jasmine, cardamom, and jackfruit. I was born and brought up in Kuwait in the Middle East, and this is where I spent the first 16 to 17 years of my life. Yeah, this is a picture of my family. When I was 17, I moved to India to do my bachelor's in food technology and management from the National Institute of Food Technology, Entrepreneurship and Management. During this, I had the opportunity to do a couple of internships in both Kuwait and India. Following my graduation, I had again the opportunity to move to Sweden to do my master's in food science and nutrition, during which I did a couple of projects and short jobs in a variety of different companies across Sweden. And following this degree, it's been a privilege to be able to move to Australia to do my PhD with Mike on such an interesting and diverse topic. So today, I will be talking to you about some of the work that I have done as part of my PhD, looking at exploring Australia's native legumes for novel food applications. So in 2020, the Committee of Agriculture, which is an important body of the FAO, held a session to talk about the steps that they need to take towards achieving their 2030 agenda. And in this meeting, the relevance and importance of Indigenous people and Indigenous food systems and the importance that that we need to place in learning from them and collaborating with them was really recognized, especially in terms of ensuring food sustainability and ensuring that food is accessible, available, and culturally acceptable to the larger populations of the world. In this regard, we also need to acknowledge that a lot of the indigenous knowledge, traditional wisdom is oral, in oral formats, and particularly shared through ancestral wisdom, stories, and also art forms, such as this dot painting made by a very talented artist called Emily Impichana from the Anmatiare tribe in near Alice Springs. So in this painting, which was fortunately commissioned as part of my PhD work, Emily recalls the stories that she wants to share through this painting, where she followed her grandmother as she harvested wattle seeds for their daily consumption. And you can see that there is a lot of information that we can collect from this particular piece of art, particularly that women in this particular symbolism carried sticks and walked towards wattle seed trees. And these trees were found in areas that had a good amount of heat, red sand, and dust, which are perfect growing conditions for wattle seeds. The yellow symbolizing that they grew in harmony with other plants that surrounded them, and the blue that symbolized the perfect blue skies that are characteristic of Alice Springs. And I would also like to point out the circular symbols used in this painting, which are used for the kulaman, or the dish that is used to collect and clean these seeds. And in recognizing the value that these forms of art and carvings and drawings bring into indigenous knowledge, we can really start to connect this to scientific evidence that we have been collecting as part of my PhD work. Looking at this from another perspective, as an average Australian consumer, starting from the Macas Australiana, there are plenty of products, both artisanal and expensive, but also some of them are economical, that are available in the Australian food market, ranging from chocolates, coffees, nut bars, nut mixes, muffin mixes, and even wattle seed extract. And as exciting as this is, it's also important to note that a lot of these formulations contain only about 1 to 3%, if that at all, of wattle seed. And this doesn't really take into account the amazing nutritional benefits that these seeds have to offer. Neither do they talk about the immense cultural and social heritage that are connected to these seeds. So this might get you curious and wondering what really are wattle seeds and why is it so important that we need to understand more about them from a scientific perspective. So wattle seeds actually are from the Acacia species and belong to the Fabaceae family, same as the green pea, scarab bean, and tamarind. As mentioned earlier, they are of huge significance to many indigenous communities across Australia, and the entire plant has found multiple uses across different generations, and they are found in almost every climatic condition across the Australian continent. So much so that the Acacia pycnantha is actually Australia's national floral emblem, and it's the green and gold of its foliage that provided inspiration for Australia's official colors. Additionally, acacia species from other parts of the world and some acacia species in Australia have been studied over the past couple of years and are found to be rich in dietary fiber. They're a good source of fatty acids like linoleic acid. They're a good source of elemental micronutrients like iron, potassium, calcium, and zinc. They have a good protein content that ranges from between 17 to 23%, which is comparable to a lot of conventional legumes. And they're very rich in bioactive compounds, which we now know have a lot of preventative health and weight management benefits. It is also important to note that there are around 2,000 different species of acacia found across the globe, and over 70% of this diversity is native and unique to Australia, and again has found immense significance with Indigenous communities across the country, with the 3 uses listed here being some of the more documented ones. So in my PhD, my main focus is on 4 different aspects of the wattle seed. What are their characteristic features? What are their dietary capabilities? What is the real sensory potential that we can harness from wattle seeds? And how can we select effective food matrices to ensure that we let the wattle seeds shine, both in terms of its story, texture, flavor, and nutrition? And I also try and connect all of the findings that I have to lived experiences, stories, art, and traditional knowledge that we have from our interactions with Indigenous communities, as well as documented traditional knowledge. I want to mention that we worked with 6 different species over the last over my PhD, and these 6 species were selected based on documented use in traditional in Indigenous communities, as well as seasonal availability. So because these seeds are small and really hardy, it was a challenge to try and really understand the seed structure. So using micro-CT or X-ray computed tomography, which is a non-invasive technique, we were able to establish the legume quality of these seeds, particularly that it has a dicotyledonous structure, which is characteristic of legumes. We can also notice that there is quite a lot of structural difference between the different species, especially if you look at Acacia victoriae, which has a much thicker seed coat and these granular structures on the inside, which really sets it apart from the 5 other species. Next, we also looked at elemental micronutrients, which is really important when we want to consider the nutritional impact that these seeds have. So you can see that, again, there is a lot of diversity between these 6 species, particularly if you look at iron, which is really important in this context because there is established evidence of chronic anemia in many different Indigenous communities across Australia. So it might be using wattle seeds in food products might be a promising solution. Another interesting thing to note is the good amount of zinc that we can find in certain species like the Acacia micropotria. But of course, if we want to utilize these elemental micronutrients effectively in food product development, we also need to really figure out where these micronutrients are located so we can develop processing techniques to better harness them and improve bioavailability. So to do that, we used X-ray fluorescence. And as expected, you can see that potassium and calcium were mostly concentrated around the seed coat. Zinc was mostly spread throughout the endosperm, while phosphorus and iron were mostly found together, which suggests iron and phosphorus chelated complexes. And this format and distribution is quite similar to many other legumes that we know. And there is quite a lot of in this sense between the different species. So although the content varies between species, the location is almost similar. So we can use the same techniques between all these different species to ensure improved bioavailability. So to look for other compositional differences between the species, we also used NMR. And using NMR, you can see that if you focus on protein, you can see that the Acacia victoriae has a much lower protein content when compared to the others, while Acacia adsurgens on the lower end has quite a few aromatics that sets it apart from the other species. To further quantify this, we looked at protein, dietary fiber, lipids, and starch in the 6 different species. And it's very interesting to see that, again, there is a lot of diversity, particularly if you look at protein, the range is between 17 to 27%, which is quite a lot, considering that they all belong to the same species. There is a lot of dietary fiber that is, again, from 50 to 77%, which is not characteristic of conventional legumes. And another very exciting feature of these seeds is its very variable lipid profile, which can range from 6% to 23%. And this is just a general comparison to chickpeas because it's one of the most popular legumes that belong to the same Fabaceae family. So again, focusing on the lipid content, we noticed that when we extracted the lipids, they were heavily pigmented, which led us to look for carotenoid content. And again, interestingly enough, there is a huge variability between the different species, with the Acacia victoriae containing less than 50 micrograms per gram, and Tenuissima having the highest out of all of these 6 species. Interestingly enough, when we looked at the lipid content, it was the Acacia tenuissima that had the highest lipid content. And this led us to confirm our hypothesis that it is the aril or the attachment of the seed to the pod, as you can see here, that contributes significantly towards the carotenoid content as well as the lipid profile. Another thing that I would like to note here is that the Acacia tenuissima's carotenoid content is very comparable to the carotenoid content of sweet potatoes, which is known to be beneficial, particularly for its carotenoid content. Moving on, in order to really understand the dietary capabilities of Australian native wattle seeds, we used in vitro batch gut fermentation with human fecal inoculum. We used 30 different substrates. So we processed 6 different species in 5 different ways and tried to understand the dietary capabilities in terms of gas production kinetics, end products like short-chain fatty acids and ammonia, and really followed the breakdown of the substrate during the digestion process with NMR. So the gas profiles are indicative of the level of microbial activity. And from here, you can see that for the raw undigested samples, there was not really as much of a difference as we expected. You can see that the Acacia coli, Acacia microbiota, and Acacia victoriae had a very similar gas profile when compared to the Acacia adsurgens and Acacia tenuissima. But an exciting development was that when processed, different species had completely different effects. For example, you can see here that the Acacia coli, when processed, all the different processing methods behaved almost similarly when compared to the raw and undigested seed. Whereas for Acacia tenuissima, there was quite a lot of difference between the different processing methods. And it's interesting to also note that the method that was used for all the species were exactly the same. So it is definitely the diversity between the different species that's again playing a role here. But we need to further investigate on what those differences are and how they contribute to the varying microbial activity. Next, moving on to the sensory potential, we focused on the aroma profiles of wattle seeds and the flavor profiles. So for the aroma profiles, we had 24 different samples. So 4 different wattle seed species in 6 different methods. We had whole raw, ground raw, malted and ground, dry roasted and ground, which is the traditionally used method, wet roasted and ground, and pressure cooked whole. With a trained sensory panel, we came up with 13 different aroma descriptors, and I'd like you to focus on the sweet note, described by the panel as a sugary smell like vanilla bean, cinnamon, and chocolate. The dark or dry fruit note, described by the panel as a heady, rich, and slightly sweet note. Yeasty, described as a dirty, sour smell like flat beer. And woody, described as a fragrant aroma of wood chips, cedar, and fresh sawdust. And using a scale that ranged from 0 to 100, 0 being none and 100 being high, we asked the panel to scale each of these attributes. And when we statistically analyzed the differences, 3 different clusters emerged. So you can see that the wet roasted and dry roasted were all in the same cluster with more toasted and savory notes. The whole raw seed and ground raw seed were in the same cluster with more herbal and dusty notes. And interestingly, the Acacia, all the malted seeds and most of the pressure cooked seeds were in the same cluster with more yeasty and musty notes. Another observation that we made was the Acacia victoriae, when pressure cooked, had a very different aroma profile when compared to all of the other species, with more dark fruit, sweet notes, and woody notes. And even within the same cluster, you can see that Acacia victoriae was always a little bit away from the others. So to further understand why, we looked at each of these species as an average of all of these processing methods. And you can see that the aroma pattern of each of these species is different, particularly the Acacia victoriae. So this confirms our hypothesis that it is the wattle seed variety that drives the aroma profile of processed seeds. When we looked at flavor, you can see that again, 3 clusters emerged, but each method was confined within the same cluster, and which sort of points towards the direction that processing method has a larger impact on flavor, and that there is it creates a lot more similarities in terms of the flavor profile if they're processed the exact same way. So to further understand this, we looked at a cobweb plot of each of the methods as an average of the species that we used. And you can see that, first of all, The control, which was the semolina that the wattle seeds were mixed in, did not contribute as much towards the flavor. And particularly, there is quite a lot of difference between the dry roasting and wet roasting. And the only difference in the method that we used was the addition of moisture. So you can see that wet roasting resulted in much lower burnt, bitter, and acrid flavors, and with a very similar flavor intensity profile. Next, using all of this knowledge that we collected, including the scientific evidence, as well as the stories that we've heard, as well as the uses that we know from documented evidence, we developed 3 different products, which was the muesli bar, the veggie burger patty, and the savory porridge, inspired by traditional Indigenous Australian cultural use, as well as my own cultural heritage. And these products contain 20 to 30% wattle seeds and have higher fiber, higher protein, a lower glycemic index, lower sugar, and are a better source of iron than its conventional counterparts. I'm sure we've all tried muesli bars and veggie burger patties. Probably not savory porridges, though. And then once we develop these products with the very vital help of all of the staff at the Long Pocket campus, I took these recipes and tested it at the industrial kitchen by Sheldon Catering in Darwin, where we scaled it up and further developed these recipes to fit a more large-scale process. And following this, we did a consumer study with about 126 consumers. I'm sure some of you might have been a part of this. So using the product experience as the main focus, we wanted to understand the effect of demographics, food choice behavior, shopping habits, and prior exposure or familiarity to Australian native foods, to understand the attitudes towards wattle seeds and how that influences their product experience. And during the consumer study, we also offered additional information on the socioeconomic impact, the historical relevance, geographical profiles, and nutritional information of these seeds to see if the added information had an impact on product liking and the product experience that each consumer had. So some of the key takeaways from this entire project has been to really understand that wattle seeds, to understand and establish scientific evidence on the fact that wattle seeds are an excellent source of protein, fiber. They're a good source of iron. They contain a lot of healthy fats, and they have a very low starch content, which makes them a truly unique legume. We know that wattle seed species is what the diversity between wattle seed species is what drives aroma profiles. We know that processing methods are what determine the flavor profile of these seeds. And we know that the seed addle is what contributes to the carotenoid content. So if that is our focus, it makes it easier to pick the species that fit that particular product application. But another very interesting and enriching experience through this entire PhD has been learning and working with people from different Indigenous communities. I've had the opportunity to attend the Parchama Light Festival in Alice Springs. I've had the opportunity to cook and learn from people from different Indigenous communities. I've had the opportunity to meet people like Emily, as I mentioned earlier, Raylene Brown, who is the chair of the Indigenous Bush Food Alliance, and really share my experience and my products that were developed as part of this project with them, and really see the happiness and comfort in their faces. And that's been a really heartwarming thing for me as part of this project. So if everything goes well, at the end of this work, some of the more optimistic outcomes that we have from this project is introducing all of the evidence that we've collected as part of this work as educational material into schools, both in language and in English. Using these products as a base to start more Aboriginal-owned food businesses on country, and of course, reintroducing these foods back into community to adults and children, addressing preventative health outcomes by improving affordable access to familiar and culturally appropriate food. So just to put it out there, there are a few results that are missing in this presentation. Particularly, why do different wattle seed species behave differently during in vitro gut fermentation? What contributes to such a high dietary fiber content in wattle seeds? Why did I select these particular processing methods? And are there any significant structural and compositional changes as a result of these processing techniques? And what have we really learned from this consumer study? And how can we apply that to better understand consumer markets that are more suited to such a niche ingredient? And if you'd like to know more about that, I have my thesis review on the 11th of September. So feel free to attend that. And thank you so much for your attention, and happy to take any questions.
Thank you very much, Sarah. That was excellent. So please think up your questions, put them into the Q&A function, and we'll get to them. So we have a first question from Sheila Ome. She says, good research. Are there plans to conserve the plant in situ, i.e. in nature? So what is the plan to conserve and sustain?
So a lot of the seeds that I have used in my project come from wild harvested sources, so trees that have naturally been there for the past couple of decades. And as far as I'm aware, there aren't any illnesses. Wattle seeds are generally a very hardy plant. They grow better after a fire, and there's very little that really affects them. But because of the fact that it's wild harvested, it's very seasonal, and supply is very is not as you would expect with a more domesticated species. So yeah, I'm not aware of any plans to conserve it as such.
So if I follow on from that, so you talked about domestication, and that's always been one of the routes to turning a native species into an agricultural one. What are your thoughts on whether that's either a good thing to do or a useful thing to do? Would that overcome some of the supply variability you talked about? But what would that go up against the wishes and thoughts of traditional owners?
So this is my personal opinion, but I think there is room for both, because in terms of wild harvesting, if we stick to wild harvesting, there is always going to be a problem with supply unless we can form a sort of cooperative model where different communities wild harvest and they bring their supply together, and then people interested in purchasing can purchase through that cooperative model. Another option would be to keep wild harvesting as it is, because there is a lot of history and culture and connection associated with maintaining that for indigenous communities, as far as I'm aware. But if we also have domesticated supply, then we can ensure that there is enough for premium products through the wild harvested angle, but there is also enough for more budget-friendly, affordable products through the domesticated pathway.
Yeah, it's a complex landscape to navigate that one, and I think watch this space, because there's an intrinsic tension in here between preserving traditional approaches and creating the scale that you need to make wattle seeds more than a 1% in a food product.
Yeah.
A question here on looking a bit broad, it was about geography, really. So which parts of Australia are wattle seeds mostly concentrated? And the second part is, are they domesticated in other countries?
So thank you for your question. As I mentioned, you can find them everywhere in Australia. Every state in Australia has wattle seed freely growing. and they are a very, again, as I mentioned, hardy plant. So irrespective of the weather, whether it's tropical, subtropical, or cold, you will find one species or the other. Whether they're edible or not is a different question, because we only know about 40 of these species that are edible out of the almost 1,000 species that grow in Australia. The second question, sorry,
was about
the second question. Yes, there are a few species that. There are a few species that have been domesticated in other countries, particularly. I think there is someone called Peter Cunningham who introduced a couple of species into Africa as a potential food source a couple of years ago, and that has been growing really successfully. And a lot of research has come out of that as well. Yeah.
Okay, thank you. Moving on to nutrition now, for a question from Guangli. I guess the pointy question is, are the minerals and polyphenols that you talked about, do we know anything about how well they are absorbed by humans?
Not as much as we would like to. We would like to know. So the only I know that there have been a few studies done on iron bioavailability using cell models, and that had promising results. There is another student, or well, now he's a postdoc, Adiamo, who also worked on wattle seeds, and he looked at iron bioaccessibility using cell models. So and from his results, I can tell that particularly just focusing on iron, that it is quite bioaccessible. But yes, no one has really looked at the polyphenols.
No, I guess the amount you'd need to do a human clinical trial is probably still prohibitive to actually do that.
Yeah. And I think I'd also like to mention that as much as I would have loved to do it, supply and cost has been a huge barrier to a lot of the things that I had planned as part of my work.
Can I just ask another type of question, which is, you pointed out the high protein and high fiber nature of the seeds that you were looking at. A lot of interest at the moment around plants as sources for proteins in the human diet. Do you see a future for wattle seeds in that, or where might they fit if they were produced on a larger scale?
Yes, I definitely see a future, because I think a lot of people talk about legumes as an excellent alternative protein, and wattle seeds really fit that bill. Additionally, because of all the fiber, it would contribute very, very well to satiety. So the combination of protein and fiber would make it an excellent alternative source of protein, and particularly in the Australian context. Having a native seed or legume that has so much nutritional potential is really exciting.
Okay, technical question here. I think you mentioned about the difficulty of looking inside the wattle seeds, and that you use micro CT to do that. So there's a question around what are the challenges with being able to do that using confocal microscopy.
So we have looked at using confocal microscopy. That was one of the things that was missing from this presentation. But what in terms of looking at the seed structure, what I was more interested in was the overall seed structure, the physical seed structure. And to do that, we couldn't really figure out a way to do that with confocal. But we have been able to look at the internal microstructure of the seed using confocal, and yes, we have done that, particularly on its own, and as a result of what happens to it as a result of processing, and what happens to it as a result of digestion. Yes, we've done that. Yeah.
I guess initially there's a challenge with getting any sort of section through the raw seeds, because they are particularly brittle and tough. But I guess, yeah, once they've been processed, not so difficult. Okay, let's see if we've got any more questions. Yes, okay, it's a perfectly good general question. Why are wattle seeds still relatively novel, despite their Well, the question says abundance. I'm not sure abundance is the right word. I guess they're widespread, their widespread nature. But have they ever been produced in abundant supply?
It depends. I mean, for the past, for the users that they've had in the past, there probably was enough supply, but with the growing interest, there's definitely not enough. Growing interest and the realization that they are capable of supporting a lot more applications, there's definitely not enough available for that. Yes, and it's not abundance. It's more about where it's available everywhere. It's just that either they're not harvested or not enough is available. Not enough is harvested at once to support the amount of interest that it currently has.
And another question around nutrition, whether you've considered looking at the glycemic index.
It's a good question, but and it would be really interesting to look at it, but I am a little short on time, so based on the ingredients used and based on the formulation, it is a fair assumption to make that it will be low. It has a low glycemic index, particularly because of the fiber content. Because high fiber foods are known to modulate blood sugar response and release of so I would think so, but yeah, we don't have any evidence of it as yet.
Yeah, I think that there's so little carbohydrate in there that the concept of glycemic index doesn't really have a great deal of meaning. There's very little of glycemic potential in there in the first place, because there's virtually no starch and only very low sugars. So I think they are, by definition, they're non-glycemic rather than having a low GI. A question here from you mentioned him, Adiamo. How would you classify wattle seed polyphenol content compared to other legumes? High, moderate, low?
Oh, definitely high. Yeah. Definitely high because of, and we think that it's because of the seed coat, the thickness of the seed coat, and the color that the seed coat has, which is a huge source of the polyphenol content.
Yeah, absolutely. I think that should be one of the selling points, at least for the where that seed coat is maintained through to the foods. Yeah.
Yeah.
Okay, I'm not seeing any more questions coming up, so going, gone. Okay, that's it, Sarah. Thank you so much for answering all the questions. If you could go on to the next slide, I'll introduce the next week's talk. So on the 29th, the next Coffee Science seminar from Chris Brosnan about RNA mobility for effective plant protection. So please join us for that one. I think I thanked Sarah enough, but thank you once again, Sarah, for a really excellent and well presented show. So that's great to see, and let's keep the message up for the wattle seed. And if you just move to the last slide, just a reminder of people that if you want to keep up with what's happening in the Coffee Science seminars, just visit the webpage. We'll leave that on the screen for a few seconds if you want to get the QR code, or just go to the Coffee website and you'll find the science seminar notices going up. So please spread the word, and let's all come back for the next in this exciting series of seminars. Thank you very much, everybody. Have a good afternoon.